So when we think about skeletal muscles, we can't really talk about them as individual cells, because unlike everywhere else in the body, rather than individual cells packed together, what we actually have in skeletal muscle is long fibres where the cells, at an embryological level, the cells have fused together to form these long fibres with multiple nuclei. And rather than cell division for these muscles to get bigger and for these muscles to grow throughout childhood, rather than the cells dividing, in fact, they just replicate the nuclei and therefore get bigger. So when we talked about mitosis, do you remember the very last stage was a process called cytokinesis, where the DNA material, the genetic material, had been replicated and we had two nuclei and then the cell split. Well, in skeletal muscle fibres, they get to the point where you have duplicated the genetic material and formed two nuclei, but the process of cytokinesis doesn't happen. Instead, the cell just grows longer and longer and longer. They have a uniform diameter throughout the length of it, and that's a very, very narrow, microscopic level. If you look at this picture here, this is an electron microscope diagram, so you can see how fine we're looking at here. However, they are really long. They extend the length of the entire muscle that they're associated with. So if we're talking about the thigh muscle, that single muscle fibre is going to extend from the hip all the way to the knee. In other places they might be much shorter, say for example in your eye, there's just a wee tiny wee fine muscle, and then they'd be much shorter, but they're still considerably longer than they are light, obviously. Within the muscle fibre, we have all the things that you would expect within a normal cell. Like I said, we have multiple nuclei spread along the length of it. And then we have what in a normal cell would be called cytoplasm. But in a muscle cell, we refer to it as sarcoplasm, because, as we're about to discuss, a muscle fibre contains more than just the normal organelles that you would find within a cell. It has everything that you would expect, but there's extra on top of that. And what we're talking about here is something called myofibrils. And this is the contractile machinery of the muscle fibre. It's what allows the muscle to contract and get shorter and then relax and go longer. So myofibrils. And if you look at this image at the top here, we've got 4 muscle fibres here lined up in lengthways together. But within each muscle fibre, you can see there's lots and lots of long, thin tubules that run the full length of the muscle fibre. And those are the myofibrils. They're the very thing that makes the muscle cell or muscle fibre to be able to do what it requires to do. And that's why we refer to it as sarcoplasm rather than cytoplasm, because you wouldn't find myofibrils in normal cytoplasm. Surrounding all that is everything else that you would find in cytoplasm. So we have mitochondria, lots and lots of mitochondria, because as I said earlier, the cell's really active. And we've also got some endoplasmic reticulum. In order for the cell to be able to repair itself, it needs to be able to produce proteins, replace these individual fibres. And the nuclei of the cells get pushed out to the outside, as you can see on this diagram at the top, because the cell itself, the fibre itself, is packed with the microfibrils. There's no space for the nuclei in between there, so they squish out to the sides. Each fibre might have hundreds, possibly thousands of nuclei, depending on the size of it, but they all lie on the outside. Now we can see from this diagram here, in fact if we scroll down there's a slightly better one. So you can quite clearly see here all the microfibrils that are lined up running the full length of this individual muscle fibre. But you can see we've got these individual sort of banding sections going on all the way down here at regular intervals. These are called sarcomeres. So we have a muscle fibre that runs the length of the muscle. That is the equivalent of an individual muscle cell. Within that muscle fibre, we have myofibrils running the length. And each myofibril is broken up into these individual sections that run end to end along the full length of the myofibril. And those are called a sarcomere. And it's the sarcomeres that are the individual contractile mechanisms that allow the full myofibril and therefore the full muscle fibre to get shorter and longer. And this is where things start to get a little bit more complex, I'm afraid, and there's going to be lots of terms here to remember. What we're looking at now is an individual sarcomere. So we've taken a chunk out of that myofibril and we're looking at the sarcomere. So the sarcomere runs from this black line along to this black line. So if I highlight that, just to make it clear what we're looking at. So this line down the bottom demonstrates the length of that sarcomere. If we look at a sarcomere under a microscope, so we look at it histologically, it has some quite distinctive bands and lines on it. And they represent different fibres within the myofibril that move around. So for the first thing to look at is this dark band across the middle. I want you to ignore the white line, the white gap down it for the time being. I'll explain that later. So right in the middle of each sarcomere we have a distinct dark band, okay, that runs across here. We refer to that as the A band. And this marks the part of the sarcomere that is most densely packed with thick filaments. These filaments are called myosin. And they lie tightly packed right in the middle. Either side of the A band, we have a lighter appearance area. So along here and along here, which corresponds to, as I'm sure you've worked out, along here and along here. And that appears lighter under the microscope because it's thinner, less tightly packed filaments. So we call this the I band, and there is one on each side. And those filaments, the finer, less densely packed ones, are called actin. There is some overlap at rest between the myosin and the actin fibres. So it's not a case that as the A band stops, the actin fibres stop, and as the I band stops, the myosin fibres stop. They do insert into each other a little bit in their resting state. The dark lines down either side just signify the end of a sarcomere and the start of the next. So we call that the Z line because it's Z, it's at the end. The actin fibres run between the sarcomeres. But there's a line down the middle that marks the start of one and the end of another. So I'll just put a little arrow so it's just really clear that we're talking about this line here, and similarly this line at the other line. So Z line in either end. So that's what your sarcomere looks like at rest. Thick, wide fibres in the middle, thinner, less tightly packed at either end, and with a little bit of overlap. And at rest, there's a light band down the middle, and this is this white line I've told you to ignore up to this point. And that's because there's a point in the middle where there is just myosin. We've got no overlap because the actin fibres have stopped and it's just myosin, so it looks a little bit lighter. However, when the muscle contracts, the actin fibres move towards the middle. And there is a much smaller space where there is no overlap, and histologically that white gap disappears. So we call that the H-band, and you will only see the H-band when the muscle cell is relaxed. So yeah, I'm not sure this will let me draw a curved line. It will probably straighten it up. It has, but I'm sure you know what I'm talking about there. It's the band down the middle. So just to explain that in a slightly different way, if this is a myofibril that's been cut down the middle and we're looking at the sarcomere, at the fibres that are inside it, just to make sure we match our colours. This top image is at rest, so we've got our A band running the length of the myosin filaments, and we've got our I band at either end, and that is, oh sorry, that's not in the right place. There is a little bit of overlap between the thick myosin fibres and the thin actin filaments. Sorry, there should be saying filaments, not fibres there. The Z line at either end when it's relaxed. So there's quite a big space in the middle where there's no overlap, and that's where we would see our H-band. Oh, that's very wiggly like. However, when the muscle contracts, the actin filaments are moving towards the middle, the sarcomere becomes shorter, and there is a much smaller space of overlap, and therefore the H-band disappears, and we see mostly A-band with short I-bands at either side. So I hope that makes a bit of sense there about what's happening as the muscle does its thing. It's all about the movement of these filaments towards the middle and the closing of the gap in the middle. Thinking then about how the muscle fibres get the nutrition to do this. This is obviously a really energy-rich process. Can't do it without the energy to support it. But the myofibrils are packed so tightly inside that muscle fibre that there is not a lot of space for the nutrition to get through. Therefore, we have some supporting tissue sheaths that lie around the outside of the muscle at various different points in order to get the nutrition through. So through these connective tissue sheaths run the blood vessels to deliver the nutrition and take away the waste, and also the nerves in order to stimulate the muscles to do their thing. These are voluntary muscles, remember, they need some kind of stimulation from our brain. So depending on where they are within the structure of the muscle, they've got different names. So if we start off at the very smallest point with our muscle fibre, surrounding each muscle fibre, we have a thin connective sheath called the endomysium, surrounding each individual fibre. Then the fibres group together into little bunches, which is what we can see here. They're referred to as fascicles. So we'll write that on because that is not actually on this diagram. This here is a fascicle, as are all of these individual groupings. So you've got each muscle fibre surrounded by its endomysium, all grouped together into a fascicle. The fascicle is then itself surrounded by a supporting tissue sheath called the perimysium. And then all the fascicles group together into one big group to form the muscle as a whole, which is what we can see as this whole structure. And it has a supporting tissue, connective tissue sheath around that called the epimysium. Now the muscle is fatter in the middle than it is at each end, kind of like a sausage. And the connective tissue sheaths run throughout the whole tissue, surrounding each muscle fibre, fascicle, and then surrounding the whole thing. And as the muscle gets thinner towards either end, the connective tissue sheaths come together into the thinnest point. So if you think of this like a string of sausages, and you've got a sausage surrounded by a skin, and then it comes to a thin point and a twisty bit, and which is just the sausage skin, and then it goes back to having a filling again, and then another bit again with just the sausage skin. So the supporting tissue sheaths are kind of like that sausage skin, and at either end you get to a thin point where it's just them. And at either end, that thin part that then goes on to attach to bone, so that's where the similarity with the string of sausages stops, because it doesn't go back out to form another muscle. It goes thin and it attaches to bone. That bit at either end is referred to as the tendon, which is a term that you've probably heard before. So the tendon is what attaches the muscle to the bone, and it is just these collection of supporting tissue sheaths that are around that. So just to recap, muscle fibre surrounded by endomysium. Muscle fibres grouped together into a fascicle, which is surrounded by a perimysium. And then all the fascicles group together to form the whole muscle, which is surrounded by an epimysium. And all those sheaths come together at the thin end, either end of the muscle, and they are called the tendon. So that's us kind of discussed the structure of muscle, of the fibres, and how it works. So let's think about now where the control comes from. All muscle needs control, and that comes from nerves. Whether it's voluntary or involuntary will dictate which kind of nerves supply it, and we'll talk about that more when we talk about the nervous system. However, it all needs some kind of nerve supply. And for skeletal muscle, it's controlled by the central nervous system, as with everything in our body, so our brain and our spinal cord. And then a nerve comes off from the spinal cord, as we can see here, and traverses to wherever the skeletal muscle is that we're talking about here. It could be right next door to the spinal cord and the muscles of our back, or it could be all the way down in the muscles of our feet or the muscles of our head and neck. Either way, the nerve will go to the muscle, to the individual muscle cells, and it will provide that instruction. And the term we use to describe that is innervation. And an innervation is something you're going to hear a lot throughout your time on this course, particularly when you're talking about anatomy, whether it be general or oral. And innervation simply means the nerve supply. So you might be asked a question, say, which nerve innervates this muscle, or which nerve innervates this tooth? And then you know, or simply you might be asked, what are the innervations of this tooth? You know that means which nerves supply that tooth. So for skeletal muscle, each muscle fibre will be controlled by one single nerve cell. That nerve cell may supply multiple nerve fibres. However, each nerve fibre will only have one nerve. So you don't have multiple nerves stimulating one muscle cell. That would cause issues because you'd have different instructions being given to the same cell. So one muscle cell has one nerve, but one nerve may supply multiple muscle cells. And this is what we can see from this microscopic image here. This branching tree-like structure is the nerve, one nerve. And we've got all the long, thin muscle fibres running perpendicular to it. And you can see it's got all little branches that are being all supplied by this one nerve. And when we have multiple nerve fibres being supplied by the one nerve, we refer to that as a motor unit. And that's to make sure that all these muscle fibres are doing the same thing at the same time. One muscle fibre on its own would be able to do very little. It wouldn't be strong enough, the contractions wouldn't be enough to move whatever it is that we wanted moved. However, multiple together will be strong enough, and that really depends on where in the body you are. Obviously, certain muscles, little fine muscles, so the muscles in the eye are a really classic example. Muscles that control skin movement, eyelids, things like that. They're not moving a big, huge chunk of material, so they will have multiple muscle fibres, but less than, say, for example, the thigh or the calf muscle or the bicep or the tricep, because these are moving big, heavy parts of the body. But if a muscle requires to move in the same direction, if a group of muscle cells to the same direction, they will become one motor unit and be supplied by one nerve. The way in which the nerves affect the muscle fibre to cause it to contract or relax is quite complicated and not something we need to go into a huge detail. We'll talk about it a little bit more when we talk about nervous system. However, it's suffice to say what the nerve does is it releases a chemical messenger, which is called a neurotransmitter, that causes the cell to contract. And as long as that neurotransmitter is being released, the cell will stay contracted. Once the neurotransmitter stops being released and it wears off, then the fibre will relax. So that is your innervations or your nerve supply for skeletal muscle. Different muscles, depending on what they do, will move in different ways. Some structures need multiple muscles moving in multiple directions in order to perform their functions. For example, the tongue. We don't just move the tongue in one direction. It can go in and out, up and down, but it can also get wider, it can flatten, it can move around our mouth. So it's a group of muscles that do that. And those muscles act differently, but not independently, if that makes sense. They'll all be doing something at the same time that all contributes to whatever motion it is that we want our tongue to make. So they're different motor units. They'll be supplied by different nerves, because one nerve will be saying to one muscle, right, you go wide, and another nerve will be saying to another muscle, right, you go flat, and they'll be working in conjunction, like a big team, to do the thing. And some muscles will act faster than others. We refer to that as a slow twitch or a fast twitch. So some muscles will be a really quick instant, others may take a few seconds. If you Google slow twitch versus fast twitch, you will find a really good example that's easy to understand. And when you find it, you will also understand why I'm not talking about it, but you'll see what I mean, but it's a really good example of it. So what about the energy then? We've already said that muscles need a lot of energy. Multiple mitochondria in each muscle fibre along the full length of it. And they need huge amount of raw resources in order to create enough ATP for the muscle to do what it needs to do. So we're looking here for oxygen and glucose. We have aerobic respiration going on in an ideal situation. We also need calcium, like I said earlier, and the reason why we need calcium is it helps the muscle fibres coordinate. So they're receiving this one signal and they need to all act at the same time. Calcium helps that happen. If, as adults, we are nutritionally deficient in calcium, then instead of getting a whole nerve fibre doing it at the one time, we get a sort of wave effect along the muscle fibre, and that's not ideal. So that's why calcium is important to us. And whilst all this is going on, we have a huge amount of waste that's created. We've got carbon dioxide and water created as waste products of aerobic respiration. But we've got a bit of an issue because these fibrils are so tightly, myofibrils are really tightly packed within each muscle fibre, which are then really tightly packed within the fascicles, and again tightly packed into the whole muscle. So it's quite difficult for the raw materials to get through to each individual cell, even despite the really rich blood supply that we can find in the connective tissue sheaths. And the majority of the time it works well, and that's what we're looking at here in this image. This is a microscope image where the muscle tissue has been removed and we just have the blood supply showing of the sheaths that surround each nerve fibre and fascicle and muscle as a whole. So you can see we've got lots of red lines and we've got some sort of purpley bluey colours. So the red is the capillaries, so that's the blood that's coming from the heart, high in oxygen, high in nutrients. And then the purpley bluey colour is the venules that will go on to produce veins, and that's taking away the waste blood. So the blood that's been used, taking it back towards the heart. It's low in oxygen, high in carbon dioxide because it's taking away the waste products. And that works well most of the time. However, if your muscle is working a lot for a long period of time, it sometimes gets to the point where the oxygen supply is insufficient. It still needs to keep going because it's getting messages from the brain to keep working. But it cannot get enough oxygen for aerobic respiration to happen. So instead, it switches to its second option, which is called anaerobic respiration. And anaerobic respiration is respiration that occurs out with the presence of oxygen. It's not as efficient. It doesn't allow as much ATP to be produced. And it also has a major downside in that the waste created is not just water and carbon dioxide, but lactic acid. And lactic acid is what causes the pain in our muscles when they've been working for a long time. So it's the pain you feel in your thighs when you're trying to cycle uphill, or the pain in your arms when you've been carrying your heavy shopping bags for a while. It's because the muscles are not receiving sufficient energy, not receiving sufficient oxygen, and anaerobic respiration is taking place. So I appreciate there's an awful lot of information there, a lot of new terminology that you've not heard before, and there's going to be a lot that you probably need to go back and look at. Whatever makes it easier for you, drawing diagrams, creating some kind of model. In terms of the sarcomeres and looking at the looking at the different fibres, quite a good way of thinking about it is, you know how you get straws, so you get big thick drinking straws like the ones you get in your smoothie, and you also get really little fine drinking straws like the one kids get So you put in the lid of wee cups, you know, the cups that have got the juice cups that have got the lid on the top and the straw pierces them, like a Capri Sun. So you've got your big smoothie straw and you've got your Capri Sun straw. And if you were to take a bundle of those smoothie straws and tape them together and sit them on the table, and then get a bundle of the thin straws at either end, and just leave them touching, but no more, there you've got a nice relaxed sarcomere. However, if you push the thin straws so that they move into the thick straws a little bit, they'll get shorter and it's contracted. And that's like your sarcomere when it's contracted. So the big thick the straws, the smoothie straws, are your myosin, and your little thin straws are your actin. And they're moving in and out of each other. And that's making it bigger and smaller. So there's a few different things you can do. Also, if you Google, you'll find some good videos that explain the actual process that's going on. But just to summarise really what we've talked about in terms of skeletal muscle. Rather than individual cells, we're talking about long multinuclear fibres that traverse the full length of the muscle. Each fibre contains myofibrils that pack within that fibre or muscle cell, and those house the mechanisms for which the muscle contracts and relaxes. Along the length of each myofibril, we've got sarcomeres end to end, and they're the individual units that stretch and relax and therefore alter the length of the fibre and therefore alter the length of the muscle. Skeletal muscle is under voluntary control by our central nervous system, so we can decide whether we move it or not. And because they're so active, they require a huge amount of raw material resources.